So thanks, Pravin, to invite me, and thanks to the STEM to give me the opportunity to present our work on the cell wall of Aspergillus fumigatus. So as you may know, Aspergillus fumigatus is a filamentous fungi. It's a saprophytic fungi. It's found mainly in the soil, where it's producing hyphae mycelium, and it's play an essential role in recycling of organic material. At the aerial phase, the mycelium may produce some specific organelle. We call it conidiophore, and on conidiophore there is the production of spore. We call them also conidia, and these spores are dispersed in the air to colonize a new environment. So that is the life cycle of Aspergillus fumigatus. And because we found spore in the air every day, people inhale spore, then can be found in the lung of people. And depending on the immune status of the patient, Aspergillus fumigatus is responsible for several types of disease. And particularly lung damage or chronic lung disease are responsible for the local development of Aspergillus fumigatus, such as aspergilloma, allergic disease, and also chronic aspergillosis. But the most severe form of aspergillosis occur in immunocompromised hosts. And in that case, Aspergillus may disseminate to other organs. And in that case, it's fatal in around 50% of cases. And here in Pasteur, in the Aspergillus unit, who was driven by Jean-Paul Laget, where the main topic was on the fungal cell wall, and particularly the cell wall of Aspergillus fumigatus. So the fungal cell wall is, we can see here on this image, is an exoskeleton which protects the fungal cell against external aggression. And also this exoskeleton is essential for the fungal growth, making it as an antifungal drug target. And also the polymer of the cell wall is used for the diagnosis of Aspergillus fumigatus. And also the other relation we are looking at, it because the external localization of the cell wall makes that the cell wall is the first thing that interact with the host cell, and the cell wall is able to induce or modulate the immune response during the infection. So it's for that reason we are interested in the analysis of the fungal cell wall. And we have analyzed the organization of the cell wall of Aspergillus fumigatus. The two main components, which are the beta-1,3-glucan and chitin, constitute the structural core of the cell wall. And these two polymers are found in most of the fungal species. So beta-1,3-glucan are branched through beta-1,6 linkages, making a side chain. And on the end of side chain, other polymer can be cross-linked. And here we see the example of the chitin cross-linked to the beta-1,3-glucan. And in Aspergillus fumigatus, two other polymers are also cross-linked to beta-1,3-glucan, beta-1,3,1,4-glucan, and also the Galactomannan. There is two other polymers, alpha-1,3-glucan and galactosaminogalactan, which are not cross-linked to others. And these two are involved in biofilm formation, and beta-1,3-glucan and chitin are really essential for the fungal growth. In fact, at that moment, the role of the Galactomannan is still not known. And the Galactomannan is really an unique polymer in the fungal kingdom. We schematically represent it as a linear chain of mannose with side chain of galactofuranose. And this Galactomannan is an antigen that is used for the diagnosis of aspergillosis, invasive aspergillosis. And the particularity of the Mannan chain is constituted by tetramannoside repeat unit containing 3 alpha-1,2 mannose and 1 alpha-1,6 mannoside. And the other characteristic is the localization of the Galactomannan. And we found it under 3 forms. One form is linked to the plasma membrane of the fungal cell and is linked through a particular glycolipid. We call it GPI for glycosyl phosphatidylinositol. One part of the Galactomannan is cross-linked to the beta-1,3-glucan inside the cell wall, and the last part is released in the medium. And and the function of this polymer was not known. And to try to understand its function, we'll look for enzyme involved in its biosynthesis, and particularly in the biosynthesis of the Mannan chain. And if we compare with what is known in yeast, in the model yeast Saccharomyces cerevisiae, in yeast, the Mannan are mainly linked to the N-glycan of glycoprotein, and there is a structure with an alpha-1,6 Mannan chain with a branch with alpha-1,2 mannoside. But the first step of this elongation is start with the H1, which is an alpha-1,6 mannosyl transferase, which add only 1 mannose residue. And after 2 complex, the Mannan polymerase 1 and Mannan polymerase 2 will elongate this chain, and then other mannosyl transferase, alpha-1,2 mannosyl transferase, will add mannose to make a more complex structure. And there is also other mannosyl transferase which complete the structure. All this mannosyl transferase has been also identified in the genome of Aspergillus fumigatus. So to try to understand if such enzymes are involved in the synthesis of how Galactomannan in Aspergillus fumigatus, we start to do deletion. And it was Christine in the lab who did this investigation. And she started to do single and multiple mutant. At the end, she constructed an undecoupled mutant where 11 mannosyl transferase has been deleted, 7 alpha-1,6 mannosyl transferase, and 4 alpha-1,2 mannosyl transferase. And when we look at the phenotype of this undecoupled mutant, we observe there is no difference at all with the parental strain. The same growth here in solid medium, in solid minimum medium, we did not observe any difference in growth. And also in the composition of the cell wall, there is no loss of the mannose composition, showing that we did not find any phenotype for this undecoupled mutant. And Christine was really disappointed by this result. She spent so many times, several years, to construct this undecoupled mutant, but we did not find any phenotype. But the home message we can take is we observe that enzymes that are present and share the same in vitro biochemical activity between mannosyl transferase, and the function between yeast and filamentous fungi are clearly different, even if they share the same in vitro activity. That was the first message or the first conclusion we can do from this part. And she continued with other family of alpha mannosyltransferase, and we look at the Cre2 family, and there is 3 members in Aspergillus fumigatus of this Cre2 family, which are putative alpha-1,2 mannosyltransferase, and Christine construct single mutant, and finally we get a strong phenotype. The Cre1 mutant show only a slight decrease of growth in a solid medium, but the Aktr4 and Aktr7 mutant clearly show a strong defect in filamentous growth, and which here at 50 degrees, there is quite no more growth in comparison to the parental strain. And we observe the same phenotype in liquid medium, where this 2 mutant has a defect in filamentous growth. When we look at the morphology of the mycelium, so here we can see for the parental strain, there is 2 spores here that produce linear hyphae. And when we compare with these 2 mutants, hyphae are not linear, they are branch, and we observe also some many filament for only 1 conidia, showing clearly that this absence of Aktr7 and Aktr4 gene induce an hyperbranch mutant, indicating there is a defect in apical growth in this mutant. At the cell wall level, we look at the composition, and clearly, and here it's the analysis of the alkali insoluble fraction of the cell wall, there is a strong decrease of mannose and galactose in this fraction. And this mannose and galactose correspond to the galactomannan cross-linked to the beta-1,3 glucan in the cell wall, showing that Cre4 and Cre7 are essential for the synthesis of the Galactomannan in Aspergillus fumigatus. And we produce Aktr4 as a recombinant protein to look at this activity. And in presence of GDP mannose, which is a donor of mannose, and using also the alpha-1,2 mannobiose as acceptor, we observe that this enzyme will use these 2 substrates to produce a trisaccharide. And to analyze the structure of this trisaccharide, we use a specific enzyme, which are alpha mannosidase. And here the GB alpha mannosidase, which is a non-specific, degrade totally the product and the substrate. And this product is resistant to alpha-1,6 mannosidase, but it's sensitive to the alpha-1,2 mannosidase, showing clearly that Aktr4 is an alpha-1,2 mannosyltransferase. And we look also the phenotype on the conidia, and we observe that these 2 mutants has a strong defect in the production of conidia in comparison to the parental strain. And the remaining conidia here also show some phenotype. They germinate faster, showing there is a defect in cell wall. And also the number of gem tube per conidia is increased. We get up to 7 gem tube per conidia in these 2 mutants, whereas there is only 2 or 3 in the parental strain. And even when we follow the germination of such conidia, we observe for some of them, they are able to produce several gem tubes in the same time, which is never appear in with the parental strain, showing that clearly there is a misregulation of the polarization of the germination. And just to conclude on this first part of my talk is so Aktr4 and Aktr7 are essential for the synthesis of the Galactomannan Aspergillus fumigatus, and both genes are required for the normal apical growth and polarization of the germination. And if we compare to other polymer of the cell wall, Galactomannan is the third polysaccharide which is required for the apical growth in Aspergillus fumigatus. So the two others are the beta-1,3 and chitin. And we look also at the proteins that are involved in the cell wall localization of the Galactomannan. As I already present, a part of the Galactomannan is cross-linked to the beta-1,3 glycan, suggesting that there is an enzyme involved in the transfer of the membrane-bound Galactomannan to the cell wall. And in yeast, there is two proteins, DACW1 and DFG5, that has been described to be putatively involved in the transfer on GPI anchor protein into the cell wall. And because Galactomannan, which is bound to the membrane, it's also containing a GPI anchor, we suppose that such homologue in Aspergillus fumigatus could be also involved in this transfer. And that was a work done by Laetitia in the lab, and she found there is 7 ortholog gene of DFG in Aspergillus fumigatus. 5 are well expressed at the conidial level, and during the germination, DFG3 is mainly expressed. And Laetitia construct single mutant and also multiple mutant to investigate the function of this gene. And among the single mutant, only DFG3 show a growth defect, and among the multiple mutant, only those containing the DFG3 deletion also have a growth defect. And this growth defect is associated with the hyperbranching of the hyphae, similarly to what we observe with Aktr4 and Aktr7. And at the localization of the Galactomannan, so we look the Galactomannan linked to membrane, to the cell wall, and also the Galactomannan release in the culture medium. And at the cell wall, we observe there is a decrease of the Galactomannan cross-linked to the cell wall with the increase of number of deletion until there is no more Galactomannan cross-linked to the cell wall. At the membrane, we observe there is a slight increase of Galactomannan bound to the membrane with a number of deletion, and the same with the Galactomannan release in the culture medium, showing clearly that DFG protein are required for the cross-link of the Galactomannan to the cell wall. And we observe that DFG3 plays a major role in filamentous growth, and some DFG genes are involved as for Aktr4 and Aktr7 in filamentous growth, and also in I did not get time to show that they are also involved in the conidiation. So that clearly shows that the cross-linking of the Galactomannan to the beta-1,3-glucan inside the cell wall is required for the function of the Galactomannan in Aspergillus fumigatus. And also we observe, so DFG protein are required for the transfer of Galactomannan onto the beta-1,3-glucan, but unfortunately, we did not succeed to show the enzymatic activity in vitro for DFG protein. Just to summarize my presentation on the biosynthesis of the Galactomannan in Aspergillus fumigatus. So this biosynthesis occurs in the Golgi and requires two sugar donors, the UDP galactofuranose and GDP mannose. And the UDP galactofuranose is produced by an enzyme which is the UDP galactomutase, and the absence of UGM1 lead to the absence of galactofuranose on the Mannan chain, and showing that the galactofuranose is not required for the polymerization of the Mannan chain. And there is a specific transporter of the UDP galactofuranose, and in Aspergillus fumigatus, there is 3 known galactofuranosyl transferase. So for the polymerization of the Mannan, so the GDP mannose is produced in the cytosol, transport inside the Golgi through a specific transporter. And here we know at least 2 mannosyl transferases are involved in the polymerization of the Mannan chain. And at the cell wall level, DFG is required for the transfer of the Galactomannan onto the beta-1,3-glucan. And from that data and from the phenotype, we can also say that this specific transporter and also KTR4 and KTR7 could be considered now as an antifungal target against Aspergillus fumigatus. But for instance, no inhibitor of such activity has been described yet. And to conclude, I would like to thank all members of the Aspergillus unit, particularly Jean-Paul Ladje, who many years ago started the project on the study of the Galactomannan in Aspergillus fumigatus. And thanks to all of you to listen to me, and I will be happy to take some questions.